Dear Professor Johnson, dear Early Excellence in Science Award winners, dear members of the Board of Trustees, dear Professor Winacker, dear Mr. Baumann, dear guests and colleagues, it's my pleasure to welcome you all to the Bayer Foundation Science Award ceremony here in Berlin and also around the world on the screen. Today, we are here to celebrate the achievements of 5 outstanding scientists, the 2021 winner of the prestigious Hansen Family Award, and the 4 winners of the Early Excellence in Science Awards for Biology, Chemistry, Medical Sciences, and Data Sciences. I have been very much looking forward to today's celebration. This is not only a highlight of our annual program, but after 2 difficult years due to the COVID pandemic, this is the first opportunity to come together in person with all of you. But for all of us, our days are now shadowed by the terrible and ruthless invasion of the Ukraine. For many decades, peace in Europe was simply presumed, and we became almost certain that conflicts between European countries would never be solved again by war, but rather through open and constructive dialogue. But sadly, we must face now the horror of a new war in which innocent people suffer. And when you arrived at Berlin Main Station, I think you could see the women and the children arriving here on a constant manner. And this makes us truly sorrowful. But nevertheless, we should not feel helpless, and we are certainly not speechless. With Bayer and the Bayer Foundation, we stand for democracy and condemn this unlawful invasion. Together with Bayer, we are working also on multiple ways to support the Ukrainian people as well as our employees. We do this by providing a whole package of humanitarian support, such as provision of urgently needed medicine, shelter for our employees, and as well as financial support for NGOs, as well as our key partner, the Red Cross. The vision of the Bayer Foundation is to catalyze advances in science and social innovation for a world with health for all and hunger for none. Through our programs, we enhance the impact of science as a basis for societal progress, and we empower social innovators as an enabler for long-lasting change. In line with our values, our science program specifically seeks to advance equity, collaboration, and trust in science. And our awards are a key component of this work. By identifying outstanding leaders and young talents in breakthrough areas of the life sciences, we foster interdisciplinary collaboration, which is critical to address the complex global challenges we are facing as society. We also support young scientists through international fellowships, seek to build constructive dialogue between science and society. Science is for everyone, not just for scientists. And we know that societal trust in science and new technological developments is critical if we are to solve the challenges that we are facing as humans on this planet. We are currently in the middle of a kind of a biological revolution, enabled by the fundamental discoveries in the fields such as developmental biology, immunology, microbiome research, or genomics. And it's this convergence of these fundamental discoveries in biology with novel technologies, such as cutting-edge imaging techniques, advances in artificial intelligence, new chemistry, or even quantum technologies, which is paving the way for a completely new era in science. And you will hear much more about this and this kind of convergence of technology and biology by our award winners. The Bayer Foundation has a strong history of identifying and recognizing groundbreaking new research fields at an early point in time. Since 1984, when the first outstanding scientists were recognized by one of our awards, 4 of our awardees of either the Bio or Family Hansen Award have received the Nobel Prize shortly after. Just last year, Professor Benjamin List of the Max Planck Institute for Coal Research in Mülheim and winner of the Otto Bayer Award 2012 received the Nobel Prize for Chemistry. Today, we have the pleasure of honoring 4 young outstanding scientists with the Early Excellence in Science Award and Professor Kai Jonsson with the Hansen Family Award. He receives the award for his groundbreaking work in understanding protein structure and function, as well as the development of novel techniques for protein label in living cells. And we will hear much more about this in the laudation by Professor Katrin Kramer, the chair of the board of trustees of the Bayer Foundation, and of course also by Kai Jonsson himself. To explore new fields in science and to develop new research paradigms, as well as to find new innovative solutions for societal challenges, it needs devoted, enthusiastic people with courageous minds that are driven by curiosity and passion. We as Bayer Foundation have the pleasure of collaborating and interacting with many outstanding science leaders. And it's also our goal to connect these leaders across borders and organizations and to drive interdisciplinary research and collaboration, which we feel is really crucial to advance us as a society. And with this and this interdisciplinary thought in mind, I am now very much looking forward to the award ceremony, and I'm handing over to Werner Baumann for his Keymode remarks. Thank you very much.
So, thank you, Monika. Dear Mr. Johnson, dear Mr. Kramer, dear Mr. Plischke, dear Mr. Winata, ladies and gentlemen, esteemed guests. First of all, I'd like to thank the Bayer Foundation for inviting me, because it's really great to be with you once again for a ceremony of our prestigious science awards. And particularly, it feels really good to be back physically for an event like this, such important, even though some of our guests are joining us from home. We had to cancel our award ceremony in 2020. We went to an all-virtual format in 2021, and now we do have a hybrid ceremony. So let's keep fingers crossed that we can go back to a real full physical event next time to come. Now let me start with what Monika already touched on. It was actually not only Benjamin List who was awarded the Nobel Prize, but a year earlier in 2020, it was also Emmanuel Charpentier, the former winner of the Family Hansen Award, who won the Nobel Prize. So I'd say the foundation is really up to something here in establishing something what I would call a good tradition. So Professor Johnson, no pressure on you. We keep a close eye on what's going to happen next time around when the Nobel Committee is going to sit together to identify you. So today, I would like to share just a few more thoughts with you on how we as a science-based company look at some of the amazing scientific progress that is happening while we speak. But also, quite frankly, how scientific progress is desperately needed in order to cope with the huge challenges of our time. Monika already touched on it, and I also think that before I come to this, I do need to say a few words about the really horrifying news and pictures we are currently receiving every single day from a country in the middle of Europe. As a matter of fact, war is less than 1,000 kilometers away from where we are today. Like all of you, I was not only shocked but really terrified by this aggression against people who lived in peace in Ukraine. I was born in 1962, and that was the year of the Cuba crisis. And I never thought that we would come back that close to a military conflict with Russia again, and in fact witness a war in the middle of Europe. At Bayer, and I'm sure I speak on behalf of all of you, we stand united for freedom, democracy, the rule of law, and the well-being of people. And rest assured that we are doing everything in our power to support our colleagues, their families, and the civil population at large in Ukraine. Likewise, we have also started through Monica and the Bayer Foundation to support with humanitarian aid, such as financial donations, vital medical products, and by providing accommodation to refugees. We have set up a 3 million fund for emergency disaster relief and to strengthen these emergency measures. We have already sent 2 trucks with life-saving antibiotics for up to 20,000 people to Ukraine. And it's also remarkable to see how our teams are stepping up once again. This is what Health for All, but also Hunger for None is all about. At the same time, we do see great concern that the war might continue or even spiral further out of control. And in addition to the humanitarian crisis in Ukraine, we can already see the indications of a global food crisis. Ukraine is known as the breadbasket of the world, and especially a lot of countries in Northern Africa and the Middle East are strongly relying on the import of, for example, wheat from Ukraine. And as a major agricultural company, which we are as well, we are working very hard on this right now. There's a lot of discussions going on with governments and international organizations, and we do everything we can in order to prevent a larger food crisis. One of my colleagues, as a matter of fact, the CEO of Yara, just recently said it's not about whether or not we are going to have a food crisis. By now, it's only about the extent of the food crisis that we face. But for now, the focus is really on providing rapid support in times of war and trying to prevent severe knock-on effects to many poor and already food insecure countries in the world. And this is where we are trying to contribute. Now, having said that, let me switch gears to the purpose of our event today, which is, of course, honoring excellence of science. We are doing this as a company we've doing it for many, many years. It's in the DNA of Bayer. We call Science for a Better Life our purpose, and we alternately honor outstanding research with our 2 science awards, the Otto Bayer Award and the Family Hansen Award. And actually, 2 men who are here with us today, Wolfgang Plischke and Ernst Ludwig Winnacker, have greatly and prominently contributed and advanced this tradition and appreciation of the scientific community at Bayer for many, many years. So I'm very thankful for this, and it's great that you are here with us today. Today, it's the turn of the Hansen Family Award, which goes back to Kurt Hansen, who served as a CEO of Bayer in the 1960s and 1970s. Hansen knew, as all of us do, that the success of Bayer as an innovation-driven company is directly linked to scientific progress. He always emphasized the importance of an intensive exchange with academia and particularly supported the research of young talents. It was therefore a logical step that he continued to support this by starting the Hansen Family Foundation in 1999 at the age of 89 years by donating the Hansen Family Award. Unlike me, Kurt Hansen himself was a chemist, and of course, he was also a merchant and a businessman. So he embodied the connection between science and the free market economy, between the word inside the lab and the word outside. Of course, this connection, the transformation of research into a marketable product decides about the economic success of a company like ours. But this connection is not only about making money, it's foremost about information. Scientific results not primarily lead to products and sales. It's in the first place that they lead to more knowledge and facts. Scientists discover knowledge that entrepreneurs then can build on and serve people. Our new family Hansen awardee knows this very well, I guess. Elise Kai Johnson is not only a remarkable scientist, but you also co-founded 4 companies. But of course, today is all about the excellence in science, and we are very keen to learn more about your work, Mr. Johnson, in the course of today's event. It was actually very interesting when we talked about some other stuff earlier today, that I could learn that you do have a close connection to this place here in Berlin, Mr. Johnson, and that you worked as a student here on site in 1985, which closes actually the loop for us that we already saw you then as a great talent, with multi-purpose capabilities. So, and what I've taken so far away from your research is that it is really helping others, that the tools you develop with different approaches to protein labeling have enabled broad advances in chemical and cell biology. Like fireflies in nature or little molecular spies, to use one of your metaphors, your tools are telling other scientists what is happening inside cells that they cannot see from the outside. For me, this is actually great testimony, how we look at science as a way to bring great expertise together to build ecosystems that help and inspire each other. In a similar way, our event today is not only meant as a recognition to reward past achievements, but also bring the inspiring discoveries and unique skills of truly brilliant minds to a diverse audience of scientists, innovators, and society. I believe by sharing knowledge and expertise widely, we open the door to new collaborations, to new instruments in research, and ultimately, we further enhance the impact of science. The other aspect that I've taken away from your work, Mr. Johnson, is the close connection between you as a chemist and the biological questions that you're working on. And also for us at Bayer, biology plays an even bigger role today than it did do in the past. In fact, we see breakthrough innovation happening at the intersection of biology, chemistry, and artificial intelligence. And that is what Monica already referred to, what is called today the advance of the biorevolution. The innovation in that space will more and more become important for the transformation of our company from a coal to a hybrid carbon-based business, which will help us and our customers to achieve ambitious CO2 reduction targets. It will also help us for global food systems, as we will enable farmers, small and large, poor and rich, to produce more on less land because they can adapt to a rapidly changing climatic environment. The biorevolution will also lead to faster drug discovery and a transformation in health from treating to potentially curing diseases, which is the holy grail of medicine. And it will help us in our self-care business, where we will increasingly be able to understand the microbiome and provide individualized solutions, improving everyday health. All of that is much needed when I look at our world today. Probably by the end of this year, we will welcome the 8 billionth human on our planet. This number has quite come a long way from a little bit more than 3 billion people that lived on our planet when I was born. And all people need to be supplied with health, nutrition, and good living circumstances, while we at the same time need to make sure that we no longer overstep the planetary boundaries and commit to becoming carbon neutral as soon as possible. It is particularly the science around the biorevolution that does drive my optimism that we can make this possible. That's why it's my great pleasure that actually all of our 4 young scientists whom we are honoring today are working in this space at the intersection of genomics, computational modeling, and artificial intelligence. And I now want to hand it back to you, Monica, for the Early Excellence in Science Awards. Thank you very much for your attention. I wish all of us, those in the room and those on the screen, an interesting and fruitful event. And a good rest of the day, and stay safe, everybody. Thank you.
So it's now my pleasure to hand over the Early Excellence in Science Awards to our 4 outstanding winners in the categories of medical sciences, biology, chemistry, and data sciences. Unfortunately, only 2 of them could make it here in person today, but the other 2 are joining us virtually. So great to have you all here. But first, let's have a look on the video to learn a bit more about these inspiring young scientists.
My name is Dr. Marieke Oudelaar, and I work at the Max Planck Institute for Multidisciplinary Sciences in Göttingen.
My name is Connor Coley. I work at the Massachusetts Institute of Technology in Cambridge, Massachusetts.
My name is Dasha Neludova, and I am working at the Institute of Molecular and Clinical Ophthalmology in Basel.
I am Maria Zimmerman-Kogadeva. I work at the European Laboratory for Molecular Biology in Heidelberg.
Being awarded the Early Excellence in Science Award means a lot to me. I think it's really great that the Bayer Foundation recognizes such curiosity-driven research as being relevant for health and society.
What drives our research is that at the present time, after patients are centrally blind, age-related macular degeneration still has no effective treatment, and this is something that we need to address.
What inspires me as a scientist is working together with other people and make discoveries with my colleagues and friends.
What inspires me as a scientist is the potential for the machine learning techniques we develop to help discover new therapeutic molecules without relying so much on human intuition. My most exciting professional achievement this year was starting a shared position in the Department of Electrical Engineering and Computer Science within MIT's College of Computing, which speaks to the interdisciplinary nature of what we do in research in this field.
We started our lab here at the Max Planck Institute for Multidisciplinary Sciences about a year ago, and it's been super exciting to set that up and to see our team grow and develop and begin to produce exciting new scientific data.
We are starting to optimize different features of our near-infrared technology to then get every component exactly right is our professional challenge.
Bayer Foundation Science Awards strive to honor outstanding achievements in science, which opens opportunities to talk about the work that we do to a much broader audience than scientists are typically able to.
I think to all young investigators, it means that we get more visibility for our research and also get opportunities to collaborate with other researchers in industry and academia and build a network of mentors and mentees and peers. My wish for 2022 would be to build a strong team of young scientists and create an environment where they enjoy working together and they feel excited to push the boundaries of our knowledge about the world of microbes.
My wish for 2022 is really to continue to grow and develop our team here and to continue contributing to our understanding of genome organization and gene regulation.
Yeah, so congratulations to all of you. Yeah, and applause, please. But before we hand over the awards, I would ask you a few questions to learn a bit more about you and your work. Yeah, and maybe, if I may, I start with you, Marieke. So you study gene regulation and genome organization in the three-dimensional space. I have to say, a very exciting field, again, in the intersection of molecular biology and computational methods. So you are now Lise Meitner, group leader at the Max Planck Institute for Multidisciplinary Research. I love that name. It's really fantastic, in Göttingen. After you finished a bachelor at Utrecht University, a master at the Karolinska, and a PhD in Oxford. I really have to say, really an international career. So can you tell us a bit more about your work and the vision also for your group in Göttingen? That would be great.
Yeah, absolutely. So, yeah, as you could already see, we are mostly interested in the regulation of gene expression, and then in particular, the relationship with that in the three-dimensional structure that the genome folds inside the cell. And so, taking that context into consideration, we want to understand how do you switch a gene on or how do you switch a gene off. And so we have been developing tools which we can measure the structure of the genome in the cell with more and more detail. And this is exactly what we wish to continue to do in the next years in our new workplace, so that we can understand much better how the genome folds and look at that in different contexts, so we can also really identify the mechanisms that drive this shaping of the genome and how that relates to the regulation of gene expression.
Fantastic. Yeah, my dream always would be that you can once model a cell, but I think we are still far away. But I think, Maria, you are working in that field more or less also in terms of combining computer science and mathematics to systems biology. I think you also have a very great career after studying in Moscow. You have been a PhD at the ETH in Zurich, a postdoc in Yale, and are now a group leader at the EMBL in Heidelberg. Yeah? Again, very, very impressive career. And in your work, you are analyzing the human gut microbiome and also the microbial ecosystem by using mathematical modeling. Can you also share a bit about your research with all of us? Yeah.
Sure. Yeah, so as a trained computer scientist and someone who is interested in microbes and their amazing effects of microbes in our everyday life, from planet to health, I'm really very fortunate to be able to combine these two passions in my research. And together with amazing experimentalists, we are developing computational and experimental approaches to probe microbial metabolism, understand interactions within microbial communities, now mainly focusing on the gut microbiome, and also model how this gut microbiome metabolic activity affects the host. And in the past couple of years, I was also involved in understanding microbial contribution to metabolism of medical drugs. And we took bottom-up approaches where we looked at microbial metabolism of drugs in vitro and also moved it to animal models and found that microbes actually have a vast capacity to metabolize medical drugs. And we followed up a few drugs to show that they can also affect drug pharmacokinetics in the animal model. So I hope that some of the work will eventually contribute to improved drug development and also better patient therapies for human health and improved treatment outcomes in the future.
Yeah, thank you very much. It's really great to learn more about this, and so we can have more effective drugs on the long run. Yeah, thank you very much. So I also would like to welcome Dasha and Connor here on the screen. So it's very nice, at least seeing you. And maybe Connor, I mean, we have heard about modeling and also your research at the MIT. You use machine learning also to predict the best synthetic routes of small molecules, which is really a fast and evolving field in research. You also already received a number of awards like the DARPA Riser Award or the CAS Future Leader Award. So can you also tell us a bit about your work and also where you see maybe still challenges that lie ahead of you to kind of do this in a fully automated manner, the synthesis of small molecules?
Yeah, absolutely. And I apologize that I can't be there in person, but it's very exciting to join remotely. So we've collectively seen a lot of progress in using machine learning to plan routes to new molecules, which has really helped chemists identify what molecules can be easily made and how in just a few seconds. So I think these tools are already performing at a level that is useful to many medicinal and synthetic chemists, but a lot of our ongoing challenges relates to making our predictions less qualitative and more quantitative. So being precise about the exact procedures by which we should make a molecule, what reaction yields to expect, what impurities might be formed and how to remove them. And of course, as you mentioned, how to then connect these predictions directly to robotic laboratories. So that's the direction that we're moving into now.
Yeah, fascinating. And I think it really transformed already kind of chemistry and how it's being done. So looking forward to the next level. Dasha, also very nice welcoming you here on the screen. And you have 2 bachelors in biology and medicine at the University of Auckland, New Zealand, a PhD at the Friedrich Miescher Institute in Basel, and you are now a research fellow at the Institute for Molecular and Clinical Ophthalmology in Basel. The focus of your work is to restore vision for patients with age-related macular degeneration, which is a leading cause of blindness in our industrialized world. So you use, quite interestingly, a combination of gene therapy and nanotechnology for your work. So can you tell us a bit more about your approach, how you aim to restore vision for patients with age-related macular degeneration?
Of course. Thank you very much for having me here today. Macular degeneration is a disease where central vision is impaired, but peripheral vision remains relatively preserved. Potential therapies seek to restore vision by restoring retinal sensitivity to light, but achieving this without interfering with remaining peripheral vision has so far proven pretty difficult. Remaining vision rules out most of our promising emerging technologies, including optogenetic sensors, because these technologies require bright, extremely bright visible light that saturates remaining viable photoreceptors. We reasoned that sensitizing a human retina to wavelengths that functional photoreceptors are unable to see, so this is near infrared light at around 900 nanometers, can be used to supplement deteriorating natural vision without interfering with the ability to see the visible spectrum. Inspired by infrared vision in snakes, we developed nanogenetic molecular tools designed to be compatible with remaining vision for the patient group with incomplete or regional retinal degeneration, such as macular degeneration.
Yeah, fantastic and very much needed. Thank you very much. Thank you, all of you. And I think we now come to the really fun part, which is handing over the awards. So maybe Marike, can I ask you to join me here on stage? So it's my pleasure now to honor you with the Early Excellence in Science Award 2021 for biology. And it goes to you for your outstanding contributions in the development of methods for the characterization of 3D genome organization and your studies of interactions between gene regulatory elements at single gene loci. Congratulations, and this is also a small award.
Thank you, wonderful. We take a...
Thank you very much, Marike. Very nice, yeah. So now I would like to follow with you, Connor. Unfortunately, I cannot give it to you in person, but please see it as a gesture. I hand it over here, yeah. And so the Early Excellence in Science Award 2022 for chemistry goes to Connor Coley for his pioneering work in applying machine learning to virtual screening, prediction, and automated synthesis of novel small organic molecules with therapeutic potential. So congratulations, and I'm very much looking forward to welcome you here in Berlin on the next time. So this goes through the aether to you. And now I would like to ask you, Maria, to join me here. So it's my pleasure to award the Early Excellence in Science Award to Maria Zimmermann-Kugadeva for achieving detailed insights into the metabolic interactions of gut bacteria with their human host by combining computational modeling with transcriptomics and genetic engineering. So congratulations, Maria.
Thank you.
And I think you get the award if you keep it for a second. also here.
Thank you.
Thank you very much. And now I have the pleasure to hand over the award to you, Dasha. The Early Excellence in Science Award for Medical Science goes to Dasha Nelidova for developing a revolutionary therapeutic approach to restore light sensitivity in the eye retina of patients suffering from age-related macular degeneration by combining gene therapy and nanotechnology. Congratulations. So, thank you very much. And with this, I am now handing over to Patrick Kramer.
My name is Kai Jonsson. I work at the Max Planck Institute for Medical Research in Heidelberg. I am in the Department of Chemical Biology. I am driven by curiosity. I love to investigate systems or problems where I do not know in advance what the solution might be. I also enjoy trying to develop tools that help other scientists to solve their problems. What I would say, if I would have to explain my work to a group of children, is that what we're trying to do is to create little molecular spies that go into cells and tell us what's happening inside what we cannot see from the outside. Receiving the Hansen Family Award, of course, is a great honor. We are all receptive to compliments, and to know that a group of peers has selected me as the recipient of such a prestigious prize is very touching. And I must also say that I'm happy that a chemist such as me got this prize, because it might inspire the next generation of chemists to work in this important or very exciting area of using chemistry to solve biological questions. Scientists as me, first of all, get the means from society. And so we first of all have to be grateful. At the same time, the work that we do can impact the life of our society, and then we also have to find ways of communicating this to the society. And we, as the society as a whole, have to learn to listen to each other and to solutions or problems that scientists point out. Our science is very interdisciplinary. It comprises people with a background in synthetic chemistry or molecular biology. And that only works efficiently if people work together, talk to each other, and work together as a team. And that is also one of the most gratifying things if you can be part of such a laboratory, that you have the opportunity to work with people with such diverse backgrounds and knowledges. This is very enriching for all of us, but also is key of pushing our science forward.
Last week I was very moved when I read an open letter by Russian scientists. With this letter, many courageous colleagues strongly protest against the hostilities launched by the armed forces of their country in Ukraine, and I join them in this protest. Now, given the terrible news that we receive from Ukraine, should we really celebrate today? I think yes, we should, because when we celebrate outstanding scientists, as we do now, we also uphold some of our key values: peace, freedom, and also international cooperation. We also express our solidarity with the people of Ukraine. Many of them work in our laboratories, they work in our companies, and also in the social sector. So despite the misery in our European neighborhood, I would really like to direct our thoughts towards outstanding science, and more specifically towards the fundamental unit of all life, which is the cell. Imagine you could visualize your favorite protein in a living cell. Imagine there would be a routine technique to label any cellular protein with a dye. Imagine what you can do now. You can study how your favorite protein behaves in its natural environment. A dream come true for any cell biologist. But due to the work of this year's winner of the Hansen Family Award, this is no longer a dream. Instead, many laboratories are now using the protein labeling technology that he invented. And it's therefore a great pleasure to introduce you to today's awardee, Kai Jonsson. Thank you. Kai Jonsson obtains the Hansen Award for his seminal research on protein labeling in cells that has enabled many advances, both in chemical and in cell biology. But how did he accomplish such groundbreaking work? The skies, you told me, Kai, were gray when you started here in Berlin to study chemistry in 1984. But then you heard very inspiring lectures in organic chemistry that were held by Helmut Schwarz, a colleague many of us know, and I'm extremely happy that Helmut Schwarz is here today. Welcome. Kai was so taken that he immediately decided that it is organic chemistry he wanted to pursue. And after obtaining his pre-diploma, the Vordiplom, he moved to the ETH in Zurich. He completed his studies and started graduate research with Stephen Benner. However, after a few months, Kai realized that this project that was proposed for his PhD would not look promising at all. And when he explained this openly to his mentor, he simply responded, well, then do something else. And looking back, he now says that this very unexpected reaction encouraged him very early on to think freely and to think independently. And he, from that point, enjoyed very much the freedom he had in his research. And after completing his PhD in 1992, Kai Jonsson went to sunny California, joined the group of Pete Schulz at UC Berkeley. There he worked on the mechanism of isoniazid, a drug that has been used for treating tuberculosis. Kai returned to Germany afterwards, and he first joined the University of Bochum, but soon he took an irresistible offer for a tenure-track professorship at the University of Lausanne, Université de Lausanne. And he had soon to give lectures also in French, which he mastered that challenge. It was also at Lausanne where Kai developed his highly successful approaches to visualize biological molecules inside of cells. The Jonsson lab created pairs of proteins and synthetic probes that could covalently attach to each other. Briefly, the protein is expressed in cells, and then cells are incubated with the probe, leading to a specific and stable labeling of the protein. Cells can then be imaged using high-resolution fluorescence microscopy, which allows one to follow the labeled protein inside cells. The first and best-known example of this approach is the so-called SNAP-tag technology. And this SNAP-tag, what is it? It's a 182 residue polypeptide that can be attached to the protein you're interested in, and also at the same time to a fluorescent dye. And here comes the major invention. To make such attachment possible, A natural enzyme was engineered by directed evolution. This enzyme is called AGT, or O6-alkyl guanine DNA methyltransferase. Nice name. The engineered enzyme accepts as a substrate O6-benzylguanine derivatives, which contain a fluorescent dye. You can now use the engineered enzyme and attach your protein to the dye. Kai Jonsson treasures the Nature Biotechnology paper that he published in 2003 and where he first introduced this SnapTech technology to the community. This publication is really a milestone in chemical biology, also because it triggered the development of many, many other tools that have similar strategies, use similar strategies, and tools that cell biologists now use routinely. Over the last years, many tags became available, different tags to simultaneously label several different proteins inside of the same cell. And this allows for very interesting observations because proteins are very social molecules and they like to interact in a dynamic manner inside cells. Also during this time in Lausanne, Kai, you met your partner and you founded a family. And in 2017, the whole family moved to Heidelberg, and Kai Jonsson became director at the Max Planck Institute for Medical Research, which is located right next to the beautiful Neckar River. You can see the castle up there above Heidelberg. And of course, also you have great colleagues on the Neuenheimer Feld there in Heidelberg. Kai is now interested in questions such as how to measure concentrations of metabolites in compartments of the cell, the so-called organelles, or how to follow activities inside nerve cells, which he can do with unprecedented spatial and temporal resolution. Kai Jonsson stands in line with great scientists who realize the potential of chemistry for biology and medicine. Over 100 years ago, Paul Ehrlich already developed chemical staining methods to distinguish different types of blood cells, and this enabled him to diagnose even various blood diseases. And much of this work was carried out here in Berlin at the Charité. In 1907, Ehrlich suggested that it may become possible to kill disease-causing microbes by a chemical agent that at the time he called the Zauberkugel. And such a magic bullet would be a drug that he envisioned would target a specific disease. And Kai Jonsson's SnapTech technology may actually be regarded as the Zauberkugel of cell biology. Basically a magic bullet that can target one specific protein and attach a label only to this protein. But Kai Jonsson doesn't stop here. His laboratory recently reported in Science a new type of biosensor for the quantification of an amino acid, phenylalanine, in a single drop of blood. This biosensor has the potential to improve the life of many patients suffering from a metabolic disease called PKU. Using this technique, patients will be able to monitor blood levels of phenylalanine by themselves without seeing a doctor. And with such interesting applications, it's not surprising, we already heard it, that Kai is a very successful entrepreneur and has co-founded 4 biotech companies. And when you ask Kai what drives him, he will probably say that he is curious, like any good scientist, and he has fun to invent new tools that are useful to others. We heard that in the video. His leadership philosophy is as simple as powerful. He puts trust in his co-workers and encourages them to search for unconventional solutions. And you can imagine this will certainly allow him to make best use of the creative potential in his team, and also that we can expect more innovative research from the laboratory in the years to come. With this, Kai, I would like to congratulate you very much on receiving the Hansen Family Award, and I would like to wish you all the best for the future.
It's our pleasure to hand over the Foundation Award for your seminal work establishing distinct approaches to protein labeling in living cells that have enabled broad advances in chemical and cell biology. Our heartfelt congratulations, guys.
Thank you.
Many congratulations. All the best.
Thank you.
Okay, up to you.
Okay, thank you, Patrick. Thank you, Mr. Baumann, for your very kind words. Receiving this prestigious award is a great honor for me. Thinking of the people who received it in the past, the great scientists, as well as thinking of other people who actually also would be deserving this award, and then receiving it yourself is just very touching, and I'm very, very grateful for that. I'm, of course, also grateful to the Bayer Foundation for selecting me, as I already said, and to my teachers, Helmut Schwarz is here, as well as, most importantly, my co-workers, whose ingenuity, hard work, and dedication has made all of this possible. We cannot have yet a celebration that would be appropriate for this kind of award back in Heidelberg, but we will do this as soon as the Corona crisis allows us to do so. I have now the opportunity to tell you in my own words what I'm actually doing. And as Patrick already said it much more eloquently than I will be able to do this, is what we're trying to do is to visualize biology with chemistry. And in my presentation, what I'll be trying to do is to give you a historical perspective of the work that we're doing, and then explain to you through a number of examples what it is, what we do. Chemistry has always been a driver for pushing biology forwards. And if we go back 150 years, we come to the work of Paul Ehrlich that Patrick also already mentioned. And Paul Ehrlich, early in his career, was obsessed with using dyes to stain tissues and cells and to discover new biology. And there's a famous saying of the lab mates of Paul Ehrlich who witnessed his obsession that can unfortunately not translated into English. Ehrlich färbt am längsten. And as one of the examples that the work of Paul Ehrlich, I would like to introduce to you this dye monophenylrosanilin that Paul Ehrlich at the time received from the German chemical industry that developed at the time. And then he used this monophenylrosanilin, or in short dahlia, to develop a staining procedure that allowed him to identify mast cells, a new cell type that he actually called mast cells. And I find this is a fantastic example, this manuscript. You can see here a figure from the original manuscript where he depicts the mast cell. It's a fantastic example how chemistry can be used to develop or to discover new biology. However, Paul Ehrlich probably would not have been able even to imagine now the kind of resolution that we can achieve when we now today use chemical dyes and various methods to stain proteins in live cells. And as an example, I would like to show you two movies, micrographs of cells that we labeled with different chemical probes that selectively attach themselves to various proteins in the cell. And if you just look here to the image of the movie to the right, what you'll be seeing is these red little dots. These are lysosomes that are moving along the tracks of microtubuli. And these kind of experiments reveal a lot about the inner work of a cell. And so you might ask yourself, how can you achieve such a specific labeling? How can you make sure that the dye that you're interested in attaches itself only to that particular protein? About 20 years ago, when we were facing this problem, we introduced what is now called so-called self-labeling protein tags. So let me explain to you what that is. A self-labeling protein tag is a small peptide or protein that you genetically fuse to the protein of interest. And what the role of this tag is to undergo a specific, highly selective reaction with the chemical probe that you add to the cells, such that the chemical probe covalently and irreversibly attaches itself to the tag and thereby labels the protein of interest. The specificity of the approach thus comes from the genetic engineering of your protein of interest, as well as this highly specific chemical reaction between the tag and the chemical probe. And the challenging part at the time actually was to identify chemical reactions that are selective and fast enough to achieve this kind of selective labeling in live cells in the time span of minutes or maybe also hours. So what we were looking for was a tag that undergoes such a highly unusual reaction. And the protein that caught our attention was a DNA repair protein, the O6-alkyl-guanine DNA alkyl transferase, in short AGT, that Patrick also already mentioned. And what AGT does is it repairs a DNA lesion, the alkylation of guanine. And you can see here in this cartoon a methylated guanine, that methyl being an alkyl group, that then is removed in a highly specific and fast reaction by this AGT that transfers the alkyl group to a reactive cysteine residue. What is unusual in this reaction is that the AGT is actually not an enzyme. It just does one reaction. It alkylates itself and that's it. It sacrifices itself for the good of the cell. And so if you think about this, nature evolved here a protein whose goal is to alkylate itself. If we think about this in the context of protein labeling, that sounds very much like what we are looking for. So what we did is, with a lot of engineering of the protein itself and the design of appropriate substrates, we designed the so-called SNAP-tag, which is an engineered alkyl-guanine transferase that now, instead of repairing DNA, reacts with these benzylguanine derivatives that carry a chemical probe, thereby leading to a covalent and irreversible labeling of the SNAP-tag, and thereby your protein of interest. SNAP-tag labeling is as easy as 1, 2, 3. What you have to do first is you need to genetically engineer your cell, transfect the cells that they express this fusion protein. Then you just pipette the benzylguanine molecule into the solution, which then diffuses into the cell and is snapped up by SNAP-tag. And therefore then leads to the labeling, the fluorescent labeling of your fusion protein, which you then can analyze or image through fluorescent microscopy. So it's a very easy procedure, but what this cartoon actually doesn't depict is that when we introduced the SNAP-tag and solved this first problem, we actually run into another problem. And that is that most of the fluorophores or probes that we would like to use for the labeling actually do not like to go into cells. And to show you this is I depict here a number of dyes, fluorescent dyes, that biophysicists love to use. These are dyes that have high extinction coefficient, high quantum yield, are very photostable and emit and are excited in the far red spectrum of light. However, if you look at these molecules, their spectroscopic properties are due to these extended aromatic systems. These are very hydrophobic molecules, and then to keep the molecule in solution, what you add is sulfonic acids to keep them charged and not crashing out. And these molecules are exquisitely membrane impermeable. So for us, that was always an enormous source of frustration. We developed this cool tool, but then the dyes that we really wanted to use, they just didn't get into the cells. And so that was really a sense of frustration. And it also was a call for more chemistry. So what we need is dyes that actually also are biocompatible, as we call it. We then, that was about 10 years ago, stumbled about a class of dyes, the silicon rhodamines, that provided a very interesting solution to that problem. Silicon rhodamines are shown here on this slide. And what this molecule does is it exists in a dynamic equilibrium between two forms. What you see on the left is the Zwitter ion. It's polar, and it is the fluorescent form of the molecule. This molecule, however, also can form a so-called spirolactone, which is shown in black on the right. And this molecule is non-polar, non-charged, and non-fluorescent. You might now say, Why would I want to have a dye that is partially present in the non-fluorescent form? That can only reduce my brightness. However, the advantage of using these kind of molecules is that the spirolactone is a molecule that is not charged, not polar, passively can diffuse into the cell. And then that was really where we got lucky here is the undergoing of the labeling reaction pushes the equilibrium back to the open state that introduces the fluorescence. That is, there is fluorogenicity, as we call it. The molecule becomes highly fluorescent only upon binding to its target. So you have a molecule that then when you add it in combination with various labeling methods, before it binds to its targets, doesn't really contribute to the background. When it binds to its cells, it goes to the fluorescent state and then allows you for the imaging. And I would just like to show you a few micrographs of experiments where we used a combination of different probes based on these principles with different colors that we just add to cultures of cells. They diffuse into cells. You don't even have to wash them away, and they highlight the structure that they're supposed to bind to. This kind of technology has become very popular in the life sciences because of the ease of how you apply it, as well as the quality of the images, the information that they reveal. And since the introduction of both SnapTech and these various dyes that we have made, there are hundreds of publications of other scientists that use these technologies. One of my favorite publications is this one here that describes the use of one of our probes on the International Space Station, which for a short time increased the standing, my standing versus my two sons at home, because they thought that actually what I do maybe is interesting. But jokes aside, the distribution or the impact of the tools that we have developed very much depends on the commercial availability of these molecules, because scientists, being them on the International Space Station or whatever, what they need to do is they have to be able to purchase these molecules. And I have been very fortunate, that has also already been mentioned, to be involved in the founding of 2 startup companies that commercialize the labeling technologies that my laboratory has developed. I'm extremely grateful to my partners in crime in these endeavors, and that has greatly increased the impact of our work. I would also like to show you another example of how you can use this protein labeling for not only visualizing where a protein is, but also learn something about biology. For example, how a biological structure is built. And the example that I would like to discuss with you is tracking cell wall growth in budding yeast. What you see here on the left is a cartoon of a budding yeast cell. That is, budding is the process where a daughter cell is originating and then finally will separate itself from the mother cell. What differentiates us from yeast cells, among other things, is that yeast cells have the cell wall that protects them. And that cell wall is a structure of carbohydrates and proteins covalently attached to the cell wall. And so let's take one of these cell wall proteins and provide it or label it with a tag, and then use a fluorophore to label the cell wall green. As you look at the cell, the budding process is not yet finished. So what the yeast cell then will need to do in order to finish the budding or proceed with the budding is the bud needs to grow, and it needs to make new cell wall structure that was not present in the first part of the experiment. So then what we can do is we come with another different colored dye, add it to the yeast cell now, and now this new part of the cell wall is stained red if you're using a red dye. Again, you can continue this by using a blue dye, for example, to visualize then how the daughter and the mother cell are separated, sealed off by the synthesis of cell wall and membrane structure between the two. And if you now look at the micrograph of the cartoon that I show you, what you really can see is that by using the sequential labeling with different chemicals that selectively attach themselves to a certain structure, you can really demonstrate that this cartoon, what you see there on the left, corresponds to what really happens in the cell in real time. I also like to show this example in particular because this is work that I did together in collaboration with my brother Niels Jonsson, who has been also very important for me in the development as a scientist. Okay, I could go on for hours giving you examples of protein labeling and My wife can testify to that. But I don't want to abuse your patient. I also believe that showing you all these nice images gives somehow a misleading impression of like what we actually do on a daily basis in the laboratory. And most of the time what we do is we use synthetic chemistry to make molecules. And so I provided you also a picture of one of our hoods where the probes that I have just used are actually made. These hoods doesn't look as nice as the ones I've seen today in my tour here in the bio facility. But what I want to tell you with this is it is often synthetic chemistry that drives the innovation. Chemistry as the mother of invention, not only in chemical biology, also in different other fields. And so this as a reminder of how important chemistry is. The examples that I have shown to you so far focus on cell biology. But if you think about it, the possibility of attaching chemicals to proteins allows you also applications outside cell biology, for example, molecular diagnostics. The project or the example that I would like to give you concerns a disease that is called Phenylketonuria, which is an inborn error in metabolism that leads to high phenylalanine concentrations in the blood. And if during development the phenylalanine concentration in the blood of a child are too high, that leads to intellectual disability. And so then what would you need to do as a parent or these patients is they need to have their phenylalanine concentrations regularly checked by going to the hospital, having blood drawn, and then analyze it using mass spectrometry, and then adapt their diet if needed. And if you think about it, it would be wonderful if for these kind of patients there would be a simple system available that would allow them to measure phenylalanine at home, such as you do with a fantastic glucose meter. However, such a device does not exist. And so we were interested in using the tools that we have developed to come up with a biosensor for measuring phenylalanine, potentially at the point of care. And so the outline of, or the design of the biosensor is summarized here on this slide. I do not really want to go into the details of it, but what it is in short, it is a luciferase, an engineered luciferase. Luciferases, you know, are the enzymes that are responsible for the emission from light from fireflies, for example, or deep sea shrimp when they glow in the dark. And so we took this luciferase, designed the molecule that then we specifically attached to it through one of our texts. The molecule is shown there on the bottom, such that when now your biosensor binds to the analyte, the light emitted by the luciferase is changing from blue to red. And how this is happening, you can see on the right side of the screen, you see two Eppendorf tubes in which there is the biosensor in the presence and absence of analyte, and in the presence of analyte, what you see, phenylalanine is the analyte here, is you see emission of red light, whereas in the absence is blue. And so you can quantify the concentration of the analyte by determining the ratio of blue to red light. And this you can do on a simple piece of paper by spotting this biosensor on a piece of paper, then excluding external light by using this sophisticated box that has on its top a camera that then you use to take a picture. And the color of the emitted light, the color of the spot, together with the calibration curve, tells you what the concentration of the analyte is. Such a procedure has the potential to become suitable for point-of-care applications. And I show you now how in this paper that Patrick also referred to, we determined phenylalanine concentrations in patients. That is, you do a finger prick, you take a very small amount of blood, dilute it into a measurement buffer, and then pipette it on a piece of paper, from which you then take with a camera the color of the emitted light that then you use to calculate what the phenylalanine concentration is. And gratifying in collaboration with our colleagues at the Heidelberg, the university hospital, what we could see is that this simple approach in its precision is as good as the mass spectrometry that is currently used as the gold standard for measuring phenylalanine concentrations in blood or serum samples. I have to confess, of course, that this approach here at present is not yet suitable for patient self-testing because you cannot ask a patient to start pipetting him or herself. But what we're currently working on in collaboration with colleagues from the Fraunhofer Society is to try to find ways to integrate the different steps into a single chip that then hopefully we will be able to provide to patients at some point. I also show you this example because it is an example of experiments that have their origin in basic questions in chemistry or chemical biology, but then lead to something that has the potential of having a societal impact. That is the life of the PKU patients, that then they would be able to measure phenylalanine at home. With this, I would like to come to the end of my presentation. What I was hoping to show you is that the combination of chemistry, protein engineering, as well as cell biology allows you to create powerful tools to study biology. What I was also hoping to give you the impression is how much fun that is, this kind of interdisciplinary work at the interface of chemistry and biology. The final thing then is, again, thank you again to the Bayer Foundation for having me elected for this prestigious award. Thank you again for my co-workers, for their fantastic work and working with me. That is so gratifying. I'm also very grateful to our society that invests so much in education and research. And this is just, I'm very grateful of this. And then finally, I'm grateful for your patience and looking forward to interacting with you now in the course of this day. Thank you.
Yeah, thank you very much, Professor Johnson, for this really inspiring speech and showing us the outstanding science, but also how you always translate the science into products and showing us your entrepreneurial mindset, which I think is also outstanding. Yeah. Thank you very much for joining us and for being our awardee today here. With this, I would like to close this year's ceremony. Thanks again for all our awardees for joining us here or on the online screen. And I would like to invite all of you to a small reception outside. And I also ask the awardees just to join me for a final picture here on stage. So thank you very much for being here and looking forward also to a nice discussion afterwards with all of you.